Alrighty, I guess we can get started. Okay, so thank you everyone for joining this the fourth of this Agri-informatics webinar series. A couple of quick house rules. Please keep your microphones on mute. And if you've got a question, please do type it in the chat, and then we'll ask you to ask it at the end of the lecture, where Professor Walsh will address it. So without further ado, it gives me great pleasure to introduce Professor Bruce Walsh. He's a professor of ecology and evolution biology and associate head, Department of Ecology and Evolution Biology at the University of Arizona. Professor Walsh's interests are broadly in using mathematical models to explore the interface of genetics and evolution, with a particular focus on two areas: the evolution of genome structure and the analysis of complex genetic characters. Uh, in other words, quantitative genetics. So in his lecture, he'll be giving us an overview of quantitative genetics. So thank you. Over to you, Prof. Walsh.
Good morning, everyone. Let me get my get up and going here. Uh, okay, so I'm going to basically give an overview of quantitative genetics, and there's a couple of important things to remember right off the bat. Number one, um, don't worry about details in this lecture because you'll have the detailed lecture notes. I also posted on uh, readings from a couple of my book chapters on that. So I may go through some of the mathematical details a little bit quickly because I want you guys to get the main concepts in that. So just focus on the main concepts, and then the mathematical details is something you can look up a little bit later. And throughout, I'll try to stop and emphasize where the important concepts are and kind of reiterate what those we introduced were, and we'll go from there. And if I'm a little bit jaggy, sorry about that. I just got back from Australia 2 nights ago, and my body can't decide if it's 11 p.m. or 6 a.m. It's 6 a.m. where I am right now. So anyhow, I mentioned before that, let me minimize the window here. There we go. Hang on. Yeah. Um, so there's further readings that for this lecture that can be found on the website. In particular, readings from chapters 4, 7, and 8 in our 2025 textbook. That's the one in the middle here. So I'll use some notation throughout my four lectures. What's that now? Okay, so I'll use the notation throughout my four lectures here. So WVL is just the 2015 book, Genetics and Analysis of Quantitative Traits, which is the first part of the revision of the 1998 book. And then WL Walsh and Lynch is the 2018 book. This one here has more to do with selection. This has more to do with basic quant gen. So just kind of, I'll go back and forth between those a little bit. So the basic model of quantitative genetics is simply this. When you observe a trait in nature, that's your phenotype P. And that phenotype has two pieces. It has the genetic value G, and it has an environmental value E. So the way to think about the genetic value G is if you're a member of a clone, let's say a cassava accession, or an inbred line, if I would replicate you over a large number of environments and take your average value, that's your G value, your genotypic value. Any particular plant starts with that average genetic value, and then it has an environmental value added on to that. So the question in quantitative genetics is basically, what can we say about G and E given observations on P? That's really the basic field in kind of a nutshell. We have a lot of tools now to do interesting stuff, but it's basically all based upon phenotypes and relatives. A lot of stuff with molecular markers is really, if you look under the hood, using the markers to look at relatives, not anything usually more complicated than that. So I say G is the average value of if I could clone you or a plant and take its average value, that's what G is. Now, in plant breeding, especially also in animal breeding, but more so in plant breeding, we have this idea about G by E interactions. And the notion would be that if you look at that equation up there and contrast two different individuals, G1 and G2, well, that difference between those individuals should stay constant over environments. That gap between them stays the same. That's called a lack of G by E interaction. So, for example, Line one is always 10 kilograms per hectare, higher yield than line two in any environment you look at. Now, obviously, that's unrealistic. And a lot of plant biology has to do with handling G by E interactions. So your genotype, its relative performance to other individuals in the sample may change as you change environments. We call this a G by E interaction. And we'll talk about that, a fair bit over the next series of lectures from other people and also in the in-person course in South Africa later on in the year. Let me illustrate those points with this maze data. And so it's important to look at this. This isn't quite a histogram. Let's look at P1 here. So what I have here is ear length, 5, 6, 7, and 8 centimeters. And down here are the numbers. So this is the histogram here is basically right around 6.5. If you look over here, again, so these are all individuals from an inbred line. I grow out those individuals, grow out about 60 of them. There's the average number. Take inbred line 2, same thing over here. The average value is about 17. So when you cross these two individuals and look at the F1, in the F1 you see a spread and the mean is about halfway in between. But the interesting thing is when you take the F1 and cross them, you get this spread in the F2, which shows about the same mean but a much, much higher variance. Now, these simple observations actually give us a nice overview of some of the key ideas. All individuals in P1 have the same genotype and hence the same G value. So all that P1 variation is due to variation in E. Likewise, P2, they all have the same genotypic value, and basically all of that variation that they see is the environmental variation. Notice, by the way, the environmental variation here for genotype 2 is larger than genotype 1. That's not at all uncommon. In the F1, everybody here also has the same genotype. If P1 and P2 were fixed for different alleles, for example, big Q in P2 and little q in P1, then in the F1, everyone's a big q. So all these individuals also have the same genotype. So all three of these populations you see, all that variation within each population is entirely environmental. What happens when we cross the F1? Well, now in the F1 here, everyone is a big Q. In the F2, I get some big Q, some big Q, some little q. In other words, I have segregation. Loci that were heterozygous for the trait in the F1 can segregate out and give either homozygote 1, homozygote 2, or the intermediate. So the extra variation you see in the F2, that's the addition of genetic variation. So this gives you a nice overview of these different interacting factors, the environmental effects and the genetic effects. So the big issue, of course, is how do we decouple those? How do we deal with those? Because the bottom line in breeding is we're trying to make predictions. We're taking observations on phenotypes, relatives, molecular markers, environmental conditions, management conditions, and trying to use those to predict what offspring are going to look like. And so in order to do that, we have to say a little bit more about what that genetic value G looks like. And by the way, I remind people that as we go through here, if questions arise, go ahead and type them in the chat and we'll come back and deal with those. And I may take a break through the various sections and handle that. So the first big idea to keep in mind is that the audience I'm speaking to is a collection of diverse breeders. Some people may work entirely with inbred lines, maize. Some people may work with clones, cassava. Some people might work with outcrossing populations. And in those settings of clones or inbred lines versus outcrossing lines or lines that you self cross with others, how the genotype is transmitted to the offspring is fundamentally different. If the offspring is a clone, and that either could be like in cassava, you make a clone from that, or you self an inbred line and take offspring from that. Those selfed individuals in the inbred line have the same genotype as the parents. That clonal accession has the same genotype as the founding clone. So in those cases, the entire genotypic value G is passed on. Now that's really beneficial because it means interactions that arise, such as dominance, Well, if I pass on both alleles, those interactions are passed on intact. So when you have offspring which are identical genetically to the parents, the G of the parents becomes the G of the offspring. Where the situation becomes different is when the offspring are generated by crossing or random mating. So I cross an individual from line 1 to an individual from line 2, or I cross two individuals from a non-inbred population. In that case, each parent contributes one of its two alleles at each locus to its offspring, and as a result, it doesn't pass on its G. It even doesn't pass on its G over 2. Rather, it passes on a part of the genotypic value. We're going to define that shortly. We'll call it A. And so on average, each parent passes on A over 2 of its genetic worth to its offspring. That A, we'll talk about, that's called the breeding value. But this is one of the key ideas, is when you have an offspring formed from two genetically distinct parents, each parent takes one of their two alleles at random and gives it to their offspring. And as a result, they don't pass on G to their offspring. They pass upon something different called their breeding value, and they only pass on half of it, A over 2. So we're going to flush out what that A part is. This whole idea is due to R.A. Fisher in 1918, which is the paper that founded the field of quantitative genetics. So to build up to that part they pass on, we have to start with something called the average effect of an allele. And what this means is that if I have an offspring and I give that offspring, let's say, allele 1, and the other allele that offspring get is random from the population, the average effect of allele 1 is how much above or below the population mean is an offspring that gets one copy of allele 1 and the other copy at random. And if you think about it, and we'll flesh this out in a little bit, that means the average effect of an allele depends upon the population you're crossing it to. So I could pass allele 1 on to a population from, let's say, Zambia, and then a population from Ghana. And the average effects could be different even though they're the same allele. Why? Because the alleles that are interacting with in Zambia are different from the alleles potentially, or the frequencies interacting with it in Ghana. So the key idea, and this is where we build up and how we can estimate these things without knowing any genetics, is relatives share alleles. So if the trait has a genetic basis, the more genes that two individuals share, that is, the more alleles they share, the closer the relationship, the more similar they should be in trait value. And we classify resemblance by the covariance. If you recall, that's just the expected difference of your observation from for trait 1 from the mean of trait 1 times your observation for trait 2 times the mean of trait 2, and take that average over all the traits. As that covariance in absolute value becomes larger, individuals are more similar. So for example, if the trait is determined by genetics, then identical twins or clones should be more similar than two non-identical full sibs, for example. And we'll use this idea to actually estimate these components. That was one of Fisher's key idea. So let's think about the implications of that last slide. Clones pass on their entire genotype, their genotypic value G, to their offspring. Sexual progeny, however, so I have an offspring that's formed by crossing two genetically distinct parents. Each parent passes on a single allele. So the offspring has an allele from parent 1 and allele from parent 2. Clones have both alleles from parent 1, and likewise inbreds the same thing. Both alleles are from parent 1 because they self. So we talked about the average effect of an allele. This leads to something called the breeding value, A, of an individual, and that in turn leads to something called the additive variation. And these are the foundations we're going to be working with. So for example, genomic selection attempts to predict the breeding value of an individual. We'll see why that's important in a little bit. We can then use that to predict what an individual is going to be using just, for example, marker data. So let's unpack those terms. And again, I'm going to go through a little algebra. Don't worry too much about it. You can look all these things up in the PDF of the notes, which are on the course website. Likewise, I posted chapter 4 of Walsh, Fisher, and Lynch, and that goes through these in a lot more detail. So let's call the two alleles at the quantitative trait locus little a and big A. And by the way, I apologize. Little a here denotes an allele. A down here denotes an effect. I should change. I'll change that in a subsequent slide. So I can quantify the effects of the little a, and big A homozygotes on our trait of interest by letting the difference between those two homozygotes be 2A. So I've got two alleles, and I look at the impact on trait value on average. The homozygote for allele 1 minus homozygote for allele 2, that difference is 2A. Now, if the effect of the heterozygote is exactly in between them, this effect should be C. C is just a constant. We can make it anything we want. What we really care about is the difference between the two homozygotes and where the heterozygote is relative to the average of the two homozygotes. So D here is the measure of dominance. If D is 0, the heterozygote's exactly in between. If D is positive, the trait of the heterozygote is closer to the big A. If D is negative, the trait is closer to little a. We can also parameterize that using the K notation. And sometimes we use K because it makes the algebra a little bit easier. So you'll see both K and D, and those are simply measures of dominance. And again, don't worry about the details. You look up the details. Just focus on the idea that we can quantify difference between two homozygotes as 2A, and we can quantify the dominance. Now, a lot of people are probably thinking, well, how do we actually estimate these? We're going to come to that. It turns out you don't need to estimate it, which is really remarkable. So a quick example, if the values are 10, 15, and 16, the average of 10 and 15 of 16 is 13. The A that is half the difference between big A, little a, 6 over 2 is 3. The dominance, well, the predicted value would be 13 for the heterozygote. We actually see 15. The dominance D is 2. If I use the K notation, that's 2/3. So we have this concept of average effect of an allele. And what the average effect of an allele means that if you get one copy of allele, let's say 1, and the other copy is random from the population, how much above or below the population mean are you? And to quantify that, we first of all have to ask what the population mean is. So on this first row over here, whoops, let me go back a second here. On this first row of the trait values, on this second row are the frequency of these different genotypes, assuming Hardy-Weinberg. So let Q be the frequency of little a and P be the frequency of big A. So if you take that times that, plus that times that, plus that times that, you get the mean. And I simply write it down here. That's our baseline, because we're going to then compare that to individuals that just get, for example, allele A in a random allele. So what you'll see is the mean can be broken down to contribution from homozygotes and then contributions from heterozygotes. So that's our baseline. We want the average and effect of the allele, and that's the mean. So for allele big A, it's the mean offspring value given a parent transmits allele big A. So the offspring gets big A from a parent, and the other allele that offspring gets is random from your population. So the average effect of allele big A is the mean of an offspring that gets a big A allele and another random allele. By the way, that other random allele could also be another big A, minus the mean of all the offspring. So if you work out what that is, so if the parent contributes big A, then P of the time, the other parent is going to contribute a big A. You'll get a big A. There's this average value. If you have a big A, then Q of the time, the other allele at random will be a little a. You'll get a big A, little a, and there's this value. So that's the average there of an individual that gets a big A allele from one parent and the other allele is random. If I subtract that from our expression previously here, the population mean, I get this expression. Now, the key about that expression is two things. Number one, it depends upon the difference between homozygotes A and the dominance D. But number two, it also depends upon allele frequencies. So as you'll see here, P and Q enter. And that's this concept I told you before. So for a given allele, A and D don't change. But the alleles that come in at random, these alleles here, those are functions of the population I cross to. So for example, it may be that in Senegal, the frequency of big A is 0.9, but in Burkina, the frequency of big A is 0.4. Those two alleles would have, the same allele A would have different average effects in those two populations, even though its A value and its D value have not changed. What has changes is the frequency of the other random alleles. That's the average effective allele big A. You can likewise compute the average effective allele little a. If you get a little a, then P of the time you'll get a big A and get a big A little a. If you have a little a, then Q of the time you'll get another little a and get a little a little a. And you'll notice that expression there looks very similar to that expression there. Again, it depends upon the effects of homozygotes and heterozygotes, but it also changes with allele frequencies. Okay? So that's the average effect of an allele. The effective allele big A is if you have an offspring that has a big A and another random allele, how much is that above or below the population mean? That's the average effective allele big A in that population. The average effective allele big A can change over populations. We also have something called the average effect of an allelic substitution. It simply means if I take an allele and substitute a big A with a little a, there's the average effect of big A, the average effect of little a. It's the mean change in trait value when an A individual is replaced, A allele in a random individual is replaced by a big A allele. So what are the key concepts so far? The average effect of an allele for a given population, how much a deviation in the mean of a trait is generated when an offspring gets that allele and another allele from the population at random. So number one, it's a property of a particular trait. I could look at a specific SNP. That SNP may have no average effects on most traits, but for example, it may have an average effect on, let's say, water stress. So it's a property of a particular trait, and more importantly, of a particular population. So if I have a line and it has an average effect, let's say of 3, when I cross it in South Africa, when I take that same parent and let's say cross them in Algeria, I could have a different value. So the average effect is a property of a population. So the average effect of allele will change over traits and will change over populations. The average effect of a substitution is replacing a big A with the little a. How does that change the mean? So now let's talk about the kind of a little bit more details on here. Again, don't worry too much about the math. These are things you can look up later. Get the basic idea. Basic idea so far as your phenotype has a genetic component G and an environmental component E. We're now going to talk about how we break that genetic component down further. So here's the genetic component for an individual with allele I and allele J. This is the average effect of allele I. This is the average effect of allele J. For example, that could be 2 and that could be 3. That would then predict that a 2-3 heterozygote should be 2 plus 3 or 5 units of, this is the overall mean, 5 units above mean. That's the predicted part. This deviation here is interactions between these two alleles. So the average effect of 2 means that against a random allele, I'll increase my genotypic value by 2. But a particular allele, maybe I'll only increase by 1. And that deviation, this is the model prediction, and that's a deviation. That's called the dominance deviation. So this is the predicted value based upon average effects. So we have the mean. Don't worry about that. We always measure things as deviation of the mean. And then the lack of fit from this prediction are caused by dominance deviations. So Fisher's key idea, and again, this is a point I've emphasized before, if you have a clone, you pass on G. If you offspring is created by you passing on a gamete, pollen, or an egg, you pass on only 1 or 2 alleles. So the genotypic value consists of a fraction that a parent can pass on to its offspring because they pass on single alleles and a fraction that cannot. In particular, in a diploid, parents pass on single alleles to their offspring. So there's that expression again. This is the actual genotypic value for alleles I and J. That's the predicted value. If I and J have no interactions between them, this is a predicted value. Actual value minus predicted is the dominance deviation, kind of lack of fit. So again, this is kind of emphasizing what I said before. Here's your predicted value. These average effects are population specific. So at this point, you're probably asking, okay, I can kind of see this, but for almost all traits, I don't know what the average effects are. And Even if I knew the loci were involved, I may not know the frequencies or the A and D values associated with genotypes at that locus. The amazing thing about quantitative genetics is you don't need to know those. And we'll see where that comes in a second. So again, this is just reemphasizing what I said. There's a predicted value. Your dominance deviation is your observed value minus your predicted value. And from a regression standpoint, so I'm going to regress on whether you have 0, 1, or 2 copies of allele 2, let's call it big A. So this would be little a, big A, little a, big A, big A. They're the actual genotypic values, the dots. This prediction here is based upon the expression I gave before. So the predicted value here is the mean plus twice the average effects of allele 1, mean plus average effects of allele 1 and 2, why? Because it contains 1 and 2, mean plus 2 average effects of allele 2, why? Because it contains 2 copies of allele 2. The boxes are the predicted values. The dots are the actual values. So the dominance deviations are those. And so what the mathematics does is it fits that line to minimize the squared sum of the dominance deviations. And the slope of that regression is the average effect of allelic substitutions. Again, don't worry about those details. You can look those up. So, oh, good. I misspelled section. Uh, so let me just quickly correct that. Okay. So the last several slides were the technical details. Don't be too concerned with them for now. What we need to know are this idea about average effects. Then we're going to flesh that out into a concept called the breeding or adigenetic value, which is simply the sum of average effects in you over all loci for the trait of interest. The genetic variances are how much the additive effects and dominance effects vary in a population. So we're going to now flesh out those latter 2 points. So the average effects and the adigenetic values, also called the breeding value. So for a given locus with alleles I and J, that's your average adigenetic value. I simply take the allelic effect of I and J at that locus in you. Your breeding value, or adigenetic value, big A, is the sum of that over all loci K that impact your trait. So instead of computing G, what I do is I simply sum all the average effects over the trait of interest for you. And that's your breeding value. And it turns out that's something you can estimate without knowing any genetics. I can estimate that by a cross. And the breeding value is what you want because as a breeder, if you're, for example, working with clones, use experimental design as Rick has talked about, and then find those individuals with the highest G values. With crossing individuals, you want to cross individuals with the highest A values. And the phenotype is a good but not a perfect predictor of A. So you want to find those individuals with the highest breeding values. Why? Because they'll, on average, have the most exceptional offspring. And in particular, why the fuss over their breeding value? Well, suppose I have a pollen parent, and for my trait of interest, when I sum over all loci in that parent for the trait of interest, has a breeding value of 10. And the seed parent has a breeding value of -2 for, let's say, plant height. Well, the expected offspring height is simply average of the two breeding values plus the population mean. So here 10 -2 or 8 divided by 2. Why divided by 2? Because A is for both alleles. A parent passes on one of those two at random. So A, what it passes on is A over 2 or 5, A over 2 or -1 on average. There's a spread about that, but on average, you don't pass on G to your offspring, you pass on A over 2 with a lot of spread about that. Why that spread? If you're a heterozygote, sometimes you pass on big A, sometimes you pass on little a. So key, parents pass on only single alleles to their offspring, and they only pass on part of their G, in particular, on average, A over 2. So if I have a whole bunch of offspring from those two parents up here, then their height will vary quite a bit because of the environment. But even if there was no environment, their height would vary a fair bit, but it would vary about a mean of 4 units above the population mean, which is the average of the two breeding values of the parents. That's why we focus so much about breeding values, is because we can estimate those by making a cross. which is a pretty neat idea. Couple last points, and we'll be through the first part, and we'll see if there's quick questions. So I'm running my genotypic value here. That's the additive effect. That's the breeding value of that locus. That's the dominance deviation. I can write that basically as a mean, and taking the variance through here, since by construction, alpha and delta are uncorrelated. We talk about that in chapter 4 in the notes. I can then basically write the genetic variance as the additive variance plus the dominance variance, as we'll talk about in a second. So what are the key concepts so far? These are the key ideas. I have an allele I that impacts my trait of interest. Its average effect is what's the offspring value given it gets a copy of allele I and another random allele. How much above or below the population mean that is. That is a property of the allele in a particular population. That's the key. The added genetic value A or breeding value is a property of an individual. In you, I sum over all your average effects. That's the fraction of G you can pass on to your offspring because you pass on basically on average half of that. Why? You've got two alleles, you pass on one of those two. Then if I look at a population and can measure the breeding values for each individual, the variance in breeding values is the added genetic variation. That's a property of a population. So since additive variation is a function of A, and A is a function of average effects, all these features are population specific. If I change the population, the average effect changes, the breeding value changes, and the variation changes. Now, one of the reasons that additive variance is really critical, if your additive variance is 0, it means that on average, no individual is going to have better offspring than another. So what part of your total variation is due to variation in A is kind of a critical idea that we're going to talk about. That's something called the heritability. Also, we can estimate A or variance in A without knowing any of the underlying genetic details. A simple example for estimating A is in dairy cattle, right? Most dairy cattle selection occurs on males. Why? Because with artificial insemination, a bull can have 2 million offspring. Problem is, bull never shows milk production. How can you estimate a bull's breeding value for milk production when they don't even display the trait? Really easy. You take that bull, you cross it to a large number of unrelated females, and look at milk productions in the daughter. Milk production in the daughter is the dad's breeding value over 2, plus the average breeding values of the mom. Since it's a random sample, that's 0. So the estimate of the breeding value of the sire is just twice the deviation of its offspring mean from the overall mean. So I've estimated that right away, not knowing anything about how many genes are involved, what their frequencies are, nothing. So this composite sum is something I can estimate with the cross. And this is what BLUP does. Best linear unbiased prediction takes a bunch of individuals and the relatedness and estimates A for all of them. So a couple of quick parting points about the genetic variances. I can write the total genetic variance as an additive part and a dominant part. I can show the genetic variation is a function here. Notice that dominance comes into here. Let's go back and see why that's. So notice in this example here, there's a lot of dominance, right? Because the heterozygote is quite close to one homozygote. This is your best fit line. That's what VA captures. And you'll notice that the deviations that are left are relatively small. So even though there's lots of dominance, right? The heterozygote is really close to one homozygote. The variance in deviations is rather small. So additive variation basically tries to capture as much variation as is possible. And that's why the K term, that's where, remember, A times K is a measure of dominance D. I could write this as A plus D. So this is the expression of that. And you notice when dominance is absent, this is symmetric because P2 is 1 minus P1. Show picks that in a second. Likewise, here's dominance. Dominance variation only arises when the heterozygote is different from the average of the two homozygotes. So here's the additive variance when there's no dominance, symmetric about a half. And what this means is as the one allele is rare, one allele is common, there's not a lot of added variation. As you move so that both alleles, this is allele frequency, so both alleles become more common, the added variance increases. As you keep on moving further, so now the other allele becomes rare, the added variance goes down again. If I have complete dominance for a trait, the dominance variation is symmetric, highest value to half, but the added variation is now skewed. Okay. So that's a good place to stop the first part here. Let me go back and look at the chat and see if there are any particular questions. I've got to squint. I've got really bad eyesight, so I'm gonna take my picture off so you don't see me looking like this. Um,
Okay, if you have any questions, please do type them in the chat or raise your hand and
yeah,
we'll ask.
or you can go ahead and raise your hand. I can only see one slide at a time, but and again, we're about halfway through the lecture, but these are some of the key concepts. So where you're thinking about, the key concept is, as a parent, you only pass on one of your two alleles to the offspring. So what you pass on is the average effect of an allele. That allele given to an offspring and another random allele, the mean of that offspring minus the overall mean is the average effect. If I sum the average effects in you, I get your breeding value. We can estimate that with a cross. The breeding value tells how good you are as a parent because your offspring are going to have A over 2. So if your A is negative, your offspring are below the mean on average. If A is positive, depending upon your other parent, your offspring are above the mean on average. Then the variation in breeding values in a population is the additive variation. Those are kind of the key concepts so far. Again, a lot of details. Don't worry so much about those. Think about those key central ideas. Then you can go back and read, for example, chapter 4. We talk about these things in a bit more detail, go through some of the algebra. Don't see any questions. Enjoying the lecture so far. Thanks, Nelson. Good luck on your game coming up. Uh, Nelson and I are in constant contact about the World Cup for some strange reason. Okay. So I told me, give me a second. Let me put my image back on. Stop share. Turn my video back on. There we go. Okay. Now go to share. Okay. So in the first part, there we are. Zoom always wants to put the menu bar right where you need to click the buttons. In the first part, we talked about these concepts of breeding value and additive variation. Now, how do we actually measure those, right? How do we actually measure those? Well, the way we do that is by looking at resemblance between relatives. Give me a second here. Didn't mean to do that. Give me half a second here. Let's go to share. Okay. So how do we actually estimate the added variation? Well, Fisher's second key idea is the resemblance between relatives is critical. So a couple of important summary measures first, then we'll talk about how we look at relatives and use that to estimate VA and VD, the additive and dominance variation. So the heritability is a central concept in quantitative genetics. And there are two versions depending upon if you work with clones or you work with outcrossing populations. The narrow sense heritability, little h squared, is the ratio. So V of A is the additive variance. So I also write that as VARV. V of P is the phenotypic variance. The heritability is simply the fraction of variation due to additive genetic values. So a typical trait has a breeding value of, let's say, 0.2. It means that 20% of all the variation you see is due to differences in breeding values between individuals. Height has a heritability in humans after you sex correct of about 0.7 to 0.8. So that's the narrow sense heritability. And we can measure the phenotypic variance directly. We've got a bunch of plants. I simply measure them, take the variance. The breeding values, though, must be estimated. So if each plant had a little tag on them with their breeding value, I could then estimate the variance of those and estimate the breeding value directly, variance and breeding value. But I can't do that because individuals don't display their breeding values. So to estimate VA, I require a known collection of relatives. I'm going to come back to that in a second. So narrow sense is added divided by total. If you work with clones, You often use the broad sense, capital H squared as opposed to lowercase h squared. And this simply is the total genetic variation divided by the phenotype. So the difference is broad sense is always bigger than narrow sense. Why? Because VG has VA plus VD and other terms in there. And so this is the fraction of genetic variation that's in the population. Narrow sense is the fraction of variation you can exploit by crossing parents. So broad sense heritability is often used when people are trying to select among the best inbred lines and they clone those inbred lines. Narrow sense is used when people are trying to improve a population. Quick comment about broad sense heritability. So in a plant population, so what I have is I've got genotype I in environment K, then I've got the Pth plot or Pth replicate of that in that environment. And then there we are. And then in that plot, I've got the Lth individual. So a single observation has the genetic value, the environmental value, the G by E, and then a plot specific effect and a further noise from individual within that particular environment. So when you measure narrow sense heritability, this VP is based upon measuring a single individual. Down here, this BP is based upon measuring an average sample of replicate genotype I over populations. So in particular, that's the variance, that's the VP that goes in here with broad sense. Notice that depends upon the environmental design. So I'm looking at E environments, I'm looking at R replicates per environment and N plants per that. So notice that even if these variance components all stay the same, as my design changes, this variance gets smaller as I make E, R, or N larger. Why? Because these terms get small, these noise terms get smaller. And so when I change the environmental design, VP changes. If VP changes, then broad sense heritability changes. So to compare broad sense heritability, we need to use a consistent design. If you and I have soybeans measured in the same environments, we have to A, have the same number of environments, B, the same number of replicates per environments, and C, the same number of individuals per plot. Otherwise, this variance down here is going to differ. That will cause that to differ, and our broad sense measures will differ. This replication, by the way, is usually why you see broad sense heritability as being so high, 0.7, 0.8, because you can make them really high by making all these error terms small by replication. So with broad sense, which is the fraction of usable variation, narrow sense, fraction of usable variation, VA over VP, and broad sense, fraction of total variation, VJ over VP in mind. Now let's talk about how we actually estimate those. So Fisher's key observation is phenotypic resemblance among relatives. I've measured the trait value. That provides an indication of the amount of genetic variation in that trait. And how does that follow? Well, the kind of logical idea is the closer the relatives, the more similar their appearance. So the covariation is a measure of phenotypic strength. So the phenotypic covariation between trait, let's say, yield in individual 1 and yield in individual 2, that should become higher and higher as individual 1 and individual 2 become more close relatives. So how do I unpack that? Well, the key to unpack this is this concept called identical by descent, or IBD. And two alleles are identical by descent if I can trace an allele in you and an allele in me back to a single copy. There's a single DNA molecule in a recent common ancestor. Genetic covariances arise because related individuals are more likely to share alleles identical by descent than unrelated individuals. And down here is a picture of what you see, for example, for full SIBs. There's parent 1, there's parent 2. I've colored each of the alleles different. Now, by the way, just because one is red and one is green doesn't mean that they're different in state. They both could be big A. I'm simply tracking the ancestors. So, for example, if I look over here, this gets their copy, their dad's allele red, mom's allele blue. Look over here, this gets the green allele from dad and the yellow allele from mom. So individuals 1 and 2 have no alleles identical by descent. If we assume these alleles are unrelated, we'll get to that in a little bit, then Individual's got the red copy from dad. This has got the green copy from dad. This has got the blue copy from mom. This has got the yellow copy from mom. Again, this mom could be a big A, but the blue copy is what she passes on to this offspring, the yellow copy here. So these two sibs at this locus share no alleles identical by descent. In contrast, the last two sibs have one has the green allele from dad, the other has a red allele, but they both have the same yellow allele from mom. They share one allele identical by descent, and these two share both alleles. So individuals can either share no alleles identical by descent, making them unrelated. They can share one, or they can share two. And again, this is done at each locus. So on average, you may share, for example, 0.6 alleles identical by descent. It means if I average over all loci for the trait and compute 0, 1, or 2, the number of IDB alleles, then on average is 0.6. So we're going to use that concept then to more formally talk about how we estimate the additive variation VA and dominance variation VD. I didn't mention it, but it's mentioned in the online on the lecture notes. Sorry, chapter 4 and 7. This can be expanded to all sorts of epistasis terms. So, by the way, I should mention this second half now is following information from chapter 7, and molecular estimates are information given in chapter 8. So the idea is I can take a trait Z, measure an individual X and individual Y, and the phenotypic covariance, assuming the environments are randomized, is given by VA for that trait times a weight that depends upon the relationship of X and Y, plus VD for that trait times a weight depending upon the relationship between X and Y. So we're going to estimate what these weights are. Notice for a given pair of individuals, over all traits, those weights stay the same. So if I look at yield or height or stay green or pH sensitivity, those A and B values are the same for the same pair of individuals over the trait because they simply measure how similar the relatives are genetically. How many alleles do they share on average across the genome? VA and VD, though, would vary over those traits. So what are red? Those parts are consistent for a pair of individuals over all traits. Those are simply weights on how their individuals are related. What fraction have one allele? What fraction have two alleles? VA and VD change as I change the traits. So there's a lot of bookkeeping in here, so don't worry about the details. Again, this is all fleshed out in chapter 7. Let's look at what this covariance should be. I'm now going to focus on, notice I've gone from P to G, genotypic value of the parent, subscript P, genotypic value of the offspring, because we assume the environments are randomized. So what's the covariation between the genotypic value of the parent, genotypic value of the offspring? Well, the key here is parents and offspring share exactly one allele identical by descent. A parent doesn't pass on two alleles, it doesn't pass on no alleles, it passes on one allele. So writing the parent as its breeding value plus its dominance value, and the offspring as its breeding value plus its dominance value, we're going to let one denote the allele they share. Then the other allele that parent 1 has, I'm going to call it X. It doesn't matter. It could be another copy of allele 1. I don't care. The allele that the offspring gets from the other parent, I'm going to call that Y. Doesn't matter what it is, because these are random draws, and they're uncorrelated. So that's the allele they share. That's the allele parent passes on to its offspring. That's the other allele in the parent that's not passed on. That's the allele the offspring got from the other parent. So taking a covariance through, I get 9 terms, and it turns out all of them, if the subscripts don't match, they're 0. So 1 and a Y is random, that's 0. What about over here? D1X, D1Y, those don't match because X and Y are random. So the only thing they share is the fact they share allele 1. So what is that covariation between the average effect of an allele in X, average effect of allele in Y? Well, that turns out to be VA over 2. Why? Because VA is variance of allele 1 plus allele 2. These are independent, so that's twice VA. So the variance of a single average effect is simply VA over 2. So relatives that share one allele identical by descent share VA over 2, and you average this over all loci for the trait. So that then gives us a way to immediately estimate genetic variances. So in particular, the parent-offspring covariation is VA over 2, so twice the parent-offspring covariation. So I measure, for example, height in one parent and height in its offspring. Twice that covariance over all my set of parents is an estimate of the added variation. So right away, I can estimate these things simply by measuring traits and known relatives. And that's really the power of these approaches. You do the same sort of thing for half-sibs. And for half-sibs, you share, see that they have no chance of sharing two alleles. Half the time they share the same allele from dad. The other allele is random from mom. Half the time they don't. So they share one allele identical by descent half the time. Otherwise, they share none. And therefore, what they share is VA over 4. So 4 times the covariation among half-sibs for your trait is another estimate of VA. With full sibs, a little more involved because, as we mentioned, I'll just kind of skip through here. With full sibs, there's a quarter chance they share no alleles identical by descent. Again, in a given locus, you average this over all loci. Half the time they share one allele, they share one allele, they share VA over 2, as we've seen. If they share both alleles, that occurs a quarter of the time. If they share both alleles, they're the same genotype, and they share VA and VD. So average, 1/2 times that plus 1/4 times that. If you average those out, you get VA over 2 plus VD over 4. So if there's dominance present, full sibs, the phenotypic covariance between full sibs should be more similar than the parent-offspring resemblance. Now, if you're an animal breeder, don't get too excited because with full sibs, there's another term here called the maternal environmental effects, and that's always going to be non-zero. So full sibs are always more similar than parent offspring. I say always, almost always, even when there's no dominance in animal situations because they have a common mother, and that has a component we haven't accounted for in here. So that's the basic idea, and I can quantify this. Remember I told you about these weights? They're in front of VA and in front of VD. Well, the weight here is simply 1/2 times the chance you share 1 plus the chance you share 2. That's the weight on VA. We saw that up here. 1/2 share 1 plus twice share 2. And then the weight on VD is simply the chance you share 2. So that's the general expression. So R and U entirely depend upon relatives. So parent offspring, R is 1/2, U is 0. Half sibs, R is 1/4, U is 0. Full sibs, R is 1/2, U is 1/4. Clones, R is 1, U is 1. And you can expand this for epistasis, etc. And again, chapters 4 and 7 talk about that. So that's the basic concept. We're now going to close out by expanding upon that to the concept we actually use when we do genomic selection. And notice before, when I did this example here, I assume those two alleles were not related. That is, the parent was not inbred. Inbreeding means the two alleles in you have a chance of being identical by descent. We also assume the two moms were not related. So how do we remove those assumptions? How do we generalize that? Well, the generalization that we'll talk about is something called the coefficient of coancestry. And the idea behind the coefficient of coancestry is imagine yourself, you have two alleles. I take an allele from the same locus in me and you. I pick an allele at random for me. I pick an allele at that same locus random from you. The coefficient of coancestry is the probability that those two alleles are identical by descent. So it's a general measure. One of the really powerful ways to look at that is what's the inbreeding level? And inbreeding F is simply the probability the two alleles in you are identical by descent. So consider a hypothetical offspring from crossing parents X and Y. Well, the inbreeding level is simply their coefficient of coancestry. Why? I pick a random allele from X and a random allele from Y, and that makes my offspring. What's the chance a random allele from X and a random allele from Y are identical by descent? It's their coefficient of ancestry. Now, this tells you something really interesting. If X and Y are fully inbred and you cross them, is the offspring inbred? It's not unless the two parents have a non-zero coefficient of ancestry. So simply because a parent is inbred doesn't mean its offspring are going to be inbred. Inbreeding means that the allele you transmit is going to be identical by has some chance of being identical by descent to the allele that the other parent transmits. So there's close connections between inbreeding and coefficients of co-ancestry. We're going to flush those out now. And by the way, this theta is what we estimate from a pedigree or from marker data, and that's what we use in things like BLUP that we'll talk about later on in the course. That's a key thing we'll use in like genomic prediction as well. Um, so a couple things. Basically, the weight. So if you look at relatives X and Y, what's the expected uh, phenotypic covariance? Well, it's 2, their coefficient of ancestry times their added variation. So what that means in particular is the added variation, sorry, the coefficient, the phenotypic correlation between X and Y is twice their coefficient of ancestry times the added variation. Turning that around, I can then estimate added variation based just on that pair. It's the covariance among that pair divided by twice their coefficient of ancestry. If I have a whole bunch of relatives, I can make each of those pairwise estimates and put those together. And we call that BLUP or GBLUP. BLUP if we use a pedigree to estimate these coefficients of ancestry. GBLUP if I estimate those using marker data. So let's look at just a couple of these to show what the impact on there. Let's go, first of all, to the coefficient of ancestry of an individual with itself. What does that mean? It's actually a pretty straightforward concept. I'm looking at you. I pick an allele out from you. You have a blue and a red allele. Again, I don't care if they're both big A blue and a red. You get one from your dad, one from your mom. I then mark you've got a red allele. I put it back in, shake it up, draw an allele out again. If those two alleles are identical, the probably those two alleles identical by descent is the coefficient of ancestry. Notice here I've got an X and an X. That's X with itself. So there are 4 possibilities, right? You've got allele 1 and 2. And again, I'm just using these to denote different DNA molecules that you carry, one from your dad, one from your mom, not necessarily that the alleles themselves are different. So there's 4 things that can happen. I draw one, put it back, draw it out again. That occurs a quarter of the time. I draw 2, put it back, draw it again. That's a quarter of the time. And then half the time I draw 1, put it back, draw 2. So those are identical by descent. So that's a 1, that's a 1. So this is a quarter. So your coefficient of ancestry with yourself would be 1 plus 1 divided by 4 or 2. And then if you're inbred, if I draw allele A1 from you and I draw allele A2 the second time, if you're inbred with probability F, X inbreeding in you, they're identical by descent. So 1 plus 1 plus 2F, that whole thing divided by 4, gives the coefficient of ancestry with yourself, X with X, as 1 plus your inbreeding level divided by 2. If you're not inbred, your coefficient of ancestry with yourself is a half. If you are inbred, it's bigger than a half. And likewise, you see here, I can always go back and forth between coefficient of ancestry and inbreeding coefficients. So I know your coefficient of ancestry, I can estimate your level of inbreeding. Now, what's that expression look like here? So that's coefficient of ancestry with yourself. What about a parent and an offspring? So parent has alleles A1 and AP. That's the allele that doesn't pass on. An offspring has allele A1 from the parent, has an offspring, has an allele A2 from the non-parent. 2 alleles in the offspring, 2 in the parent. So half the time you draw out A1 in the offspring, half the time you draw out A1 in the parent, there's identical by descent. So a quarter, 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 quarter. Likewise, a quarter of the time, you'll draw allele A1 from the offspring and AP from the parent. Well, are those related? Well, if the parent is inbred, then A1 and AP are identical by descent. So if the parent is inbred, you get an F of P for the inbreeding level of the parent. What about over here? Allele, this is the allele from the other parent. Well, this is the draw an allele from the other parent, draw an allele from your parent. That's the coefficient of ancestry between the parent and we call the non-parent, the parent you're not focusing on. So this is also the inbreeding level in the offspring. So flushing that in. Remember before, this is a quarter. If the parent was not inbred and the parent and the other parent you're crossing it to are not related, that means the offspring is not inbred. There's that general expression. That's coefficient of co-ancestry. How related are the two individuals you're crossing? I can write that in terms of inbreeding. So the parent offspring uh, coefficient of co-ancestry goes from a quarter to 1 plus inbreeding the parent plus inbreeding the offspring divided by 4. The bottom line is when you have inbreeding, resemblances increase. You can write similar things down for full SIBs. And again, I won't go through that. The notes cover that. But for example, this would be inbreeding, for example, this would be the offspring being inbred. And the bottom line from these expressions, again, this is bookkeeping. You look up these details. The key idea is the coefficient of coancestry, theta, is for two relatives. I draw out a random allele from one, put it back, draw out a random allele from the second one. The coefficient of coancestry is the probability those alleles are identical by descent. Twice the coefficient of coancestry is the weight on added variation that gets contributed to the phenotypic covariation between those two traits. And the coefficient of coancestry between two individuals is level of inbreeding in their offspring. Key, inbreeding makes relatives more similar. So these values we had before for weights on additive and dominance variation, those weights become larger if there's any sort of inbreeding. And the expressions I showed you before shows you how much larger those weights are. Again, you look those up. Those are in chapter 7. So just about done. A couple of bookkeeping things. Again, chapter 7 talks about this entirely. If you have a pedigree, you can work down the pedigree and compute what these coefficients of ancestry are. And it's simply something called the counting method. And there's kind of the algorithm for it. I won't go into the details, but it's very easy to do. And so if you have a known pedigree, what you assume is individuals at the top of the pedigree are unrelated and not inbred. And likewise, individuals like a parent that you don't know, you assume that parent is also not inbred and unrelated. And with that, you can go through and then fill out the entire pedigree and get an entire table of all the pairwise relationships. And that's something, for example, we use in BLOP or GBLOP or genomic selection. Now, those values on a pedigree are the expected values. However, think about what we saw with full SIBs. Half the time, full SIBs share one allele identical by descent. However, a quarter of the time they share none, a quarter they share two. So on average, they share one. The pedigree value for theta would show you an average sharing of one. So that equates to, we expect 2 theta to be a half for full SIBs. That's on average. However, some full SIBs may be more similar. By chance, by random segregation, they had more pairs where they shared two alleles identical by descent. Others may be less similar. So for example, in that first case, instead of being a half, it's 0.6. In the second case, they share by chance more pairs that share no alleles. So now it's less. So you see spread about that 1/2. Some are more similar, some are less similar, on average exactly half. So a big collection of full SIBs, some are slightly more similar, some are slightly less similar. You can actually use that information. And genomic selection does that. We call those actual values the realized relationships or the actual relationships. The values of theta from a pedigree are the expected relationships. So how can we get at those? Marker data. Let's go to that in a second. Let's review the key points here. Theta, the coefficient of coancestry, is the central measure of genetic relationships, with 2 theta for a pair of relatives, giving the weight on VA, that is, what's the phenotypic covariance among those two relatives? It's 2 theta VA. Inbreeding and relatedness all inflate theta over outbred and unrelated relatives. With a known pedigree, the expected value of theta can be computed, very simply, computational procedure. As we'll now detail, if I've got dense marker data, let's say a couple of thousand SNPs, I can estimate the realized value. So I may not have a pedigree at all. Or if I do have a pedigree, individuals I call as full SIBs, some may be more similar than others by chance. So how do we estimate, finally, theta from marker data? Well, it's a concept called molecular coancestry. And here I'm ignoring identity by descent and talking about what's called identity by state. So on the top here, I've denoted the genotypes as 111000. I'm going to call one allele one SNP the reference SNP, going to score it 1. The other SNP, the non-reference SNP, that's a reference SNP homozygote. That's a heterozygote. That's a non-reference SNP homozygote. Same thing over here. So if I go ahead and do the coefficient of ancestry for this parent and that parent, I always draw an allele 0 here, allele 1 here. There's no chance they're identical by descent. Over here, always draw to 1, always draw to 1, 100% chance. Over here, half the time, I always draw to 0, half the time I draw to 0, I get that. So this is the molecular co-ancestry. So simply given the SNPs, homozygote, heterozygote, other homozygote, I can go through and score those. So basically, here is the SNP scores, individual X, individual Y. I then look up in the table. Let's see, that's a 0, 0, and a 1, 0. That's half. So taking the average overall here, the average estimate of the molecular coancestry here is 0.65. So you see the basic idea by seeing how many SNPs are shared, I can compute a molecular coancestry. Now, that's not the whole case. So let's look at this individual here. They both share a 0, 0 homozygote at SNP2. At this other SNP, they share a 1, 1. Well, suppose that the frequency of SNP0 is very high. Well, random individuals are going to share, are going to be, nice radio. Don't forget to mute. Thank you. Random individuals over here have some chance of being sharing the same alleles. So if allele 0 is frequent, that's not unusual for unrelated individuals. If allele 1 here is really rare, that's a big signal these individuals are related. So simply using this information doesn't allow me to go from a molecular coancestry to a actual coancestry because I've got to weight these by the frequencies of the alleles. And that's the key part. And chapter 8, which you have a copy of, has a much more detailed discussion about this. But the basic idea is if alleles are common, it's not surprising individuals that are unrelated share them. If alleles are rare, individuals sharing rare alleles are indication that they're likely relatives. And there's several different ways you can count this. So here's one method. So just simply says if I've got alleles A, B, and X, and C, D, and Y, this says are A and C similar, 1, 0, 1, 0. So this is basically what fraction of the pairs are identical in state. That gives me an estimate here in molecular coancestry. How do I adjust for that? Well, this is the expected value given I have random mating. So it's inflated if individuals related, if individuals are not related. So if relationship is 0, this is the chance that they share those, and that's given by Hardy-Weinberg. So this is a correction for these sharing scores by allele frequencies. And what you see is if the shared allele is common, this is pretty large. If the shared allele is rare, this is unusual. And so this is that estimate here. Notice the difference. There's no correction down here. Now there's a correction for allele frequencies. And this is the basic idea. You basically take a estimate of molecular similarity, and then you basically then average those by what the effects would be for sharing alleles under random mating. And there are several different ways to do that. I'll just show them here briefly. All those are discussed at length in chapter 8. Okay, so let me stop the share and conclude with kind of an overview. So the overview you have, let me put myself back on here. So the overview you have is that your phenotypic value is a genetic value and an environmental value. We can break that genetic value into another piece called the breeding value. Breeding value is important because on average you pass half of that on average to your offspring. We talked about how you can estimate the added variation by looking at how many alleles you share, and we talked about a general measure of allelic sharing called the coefficient of coancestry. Okay? So that's a quick overview. Again, think about the key ideas, not the details. Now go back and read 4, 7, and 8. 8 is a bit detailed. In all these chapters, there's more stuff than I talked about, but you can read them for some of the key points. 8 is fairly technical, but it gives you a breakdown about how you can estimate theta from molecular data and some of the controversies involved in that. Okay. So with that, I'll open it up to any questions people might have. And I'm a little bit late, but not too bad.
Great. Thank you so much for that, Bruce. If you have any questions, please put them into the chat or you can raise your hands and we can ask them. Bruce, we have a question from Paul Gibson in the chat. Paul, would you like to unmute and ask your question?
Yes, thank you. Uh, Actually, it's more efficient to read it in the chat than it is for
Okay.
me to speak it.
I'm gonna I'm gonna turn my video off so you don't see me staring at the screen with my bad eyesight here. Let's see. I assume you didn't ask about the certificates of attendance. Um, there we are. Ah, many of our crosses are with a small number of selected parents. Can you explain how the effects represent can you explain how the effects represent different ideas than if parents consider random sampling pump? Yeah, Paul, that's a great question, by the way. Good to speak with you again. I think it was Uganda many, many, many years ago. Enjoyed that quite a bit. My wife did too. And especially the hospitality your wife showed taking you around. Um, so so imagine you have line crosses. This is a diallel analysis. So I've got 5 line crosses. I want to do pairwise crosses. When you move to line crosses, you use something called general combining abilities, GCA, and specific combining abilities, SCA. And a GCA is basically a breeding value divided by 2. So a GCA would say for my given set of lines, let's say I've got 10 lines, when I cross line 1 to any other line, how much above or below the population mean is that line expected to be? That's the GCA. So if I cross lines 1 and 2, the mean offspring should be the overall mean plus GCA for line 1 plus GCA for line 2. Then there's an extra part, the deviation, the specific combining ability. That's, for example, where things like heterosis comes in. So these ideas about breeding values and dominance can be extracted where you have a very small number of parents, like in a diallel, where you have a specific small number of lines. So I didn't talk about that, but it extends very naturally to those settings as well. Did that clear that up, Paul? Or do you want to make some additional points on that?
Well, it works fine for GCA and SCA, but then there's a tendency to extrapolate that to variances, and that doesn't seem legitimate in small number of fixed parents.
Yeah, so that's a great point, and the key there is that what you're working with is basically kind of a fixed effects interpretation. So if you have a small number of parents, you're simply looking at the variation among the means in that sample. You're not extrapolating it to a larger sample. Other questions? And again, I remind you that we're going to then turn around and talk about selection. We'll have an overview of matrices. I think that'll be useful for a lot of people. And then we're going to talk about selection in my last 2 lectures from here. So any other general questions or comments? But before I do, let me just check here and see how I should. So good luck to Egypt and, of course, everybody's favorite team, Cape Verde, and, of course, the Black Stars. So we have 3 African teams playing today. Hopefully, Africa will continue its great run in the World Cup, but fingers crossed. And Nelson, good luck on England. They're going to need it. They look pretty bad the last couple of games.
All right, great. If we have any questions, please do raise your hands or type it in the chat.
And again, I remind you, it's on these, just think about the key ideas. Go back and read now. You've seen the key ideas. I think it'll help you out a fair bit.
Okay, great. If you do have any questions and you do think about any questions, please join us on the 6th of July, Monday, for lecture 6 on quantitative genetics, single trait selection. I've just pasted the registration links there. So that's lecture 5 on Monday, the 6th of July at 3 p.m. CAT. And from all of us, thank you so much, Professor Walsh, and look
It's
forward to
also
chatting.
Double check. I'm not sure if I do matrices first or single trait selection. I'll figure it out, but
Single
just have,
traits as far
okay,
as I can.
perfect. No
Yes.
problems.
Great. All right. Thank you so much. See you
Okay.
guys.
Cheers, everyone.
Thank you. Bye.